Yesterday, our speaker began with an introduction to her area of research. Today, we're going to pick that ball up and run with it with Lecture 2. Today, we're talking about fluorescent sensors to study metals in health. Would you please make extraordinarily welcome on a final ISS Friday morning, Professor Liz New.
Thanks, everyone. So what I talked about yesterday was a general area of chemistry called chemical biology. So that's about developing chemical tools to let us study biology. What I'm going to talk about, so that's part of my research world. My other research world is the world of bioinorganic chemistry or biological inorganic chemistry. So that is understanding the interactions of inorganic species, that is metals, with biological systems. And so this is actually the world I started in, and then I was really interested in understanding it better and having better tools to understand metals in health, and so that's why I moved into chemical biology and developing fluorescent sensors. But I thought today I'd tell you a little bit about how fascinating all the different interactions are between us and metals, and then a little bit about how we're making fluorescent sensors to study this better. So just a recap from yesterday, remember fluorescence is where we have luminescence caused by the absorption of light at one wavelength and the re-radiation at a different wavelength or a different color. And we also talked about fluorescent sensors. These are responsive sensors that change their fluorescence in response to their environment, and we saw a very simple example of a responsive sensor in tonic water, which is a sensor for salt. And we'll see some more sophisticated examples of sensors today. So today we're going to talk about why metals are so important to our health, and there are lots of different metals we interact with, and they fall into different classes. Some of them are essential for us to live, and some of them are toxic and cause us to die. And then we'll talk about using fluorescence to measure the levels of metals in our body, and we can also use those sensors to measure metals in the environment. So we're going to mainly talk today about the transition metals, so that's this middle part of the periodic table. We'll briefly touch on the metals here, the alkali and the alkaline earth metals, but these are not, they have a very different interaction with our bodies. So we're going to focus on the transition metals. And transition metals have different roles in health. We have some transition metals that are essential. If we do not access them, then we will not be able to survive, but we only need to access them in very low concentrations. Then there are some metals that are toxic. And in general, the difference between metals that are essential and metals that are toxic are to do with how much we encountered them throughout evolution. So metals that are essential tend to be the smaller, lighter transition metals, which are prevalent in the Earth's crust. And the toxic metals tend to be those that only really became accessible when, thousands of years ago, we realized how we could smelt ores and access these metals. But that was not long enough evolutionarily for the body to come up with ways to deal with them. And then all transition metals have potential to be used in therapy, and so we'll talk about that as well. But starting with essential metals, so this is the biological periodic table. So these are all of the elements that we need in order to survive. And biology is obviously dominated by carbon, nitrogen, oxygen, phosphorus, sulfur, and hydrogen. They're not inorganic, they're not metals. But these blue metals here are trace biological elements. They're only present in very low concentrations in our bodies, and yet if we do not have them, we will not be able to survive. And that's what we're going to focus on today. So in a 75 kilogram body, there's about 1.45 kilograms of metal, but more than 99% of this is the group 1 and 2 metals, sodium, potassium, calcium, magnesium. The transition metals exist in very low concentrations and very low amounts. So in each of our bodies, we only have about a 20-cent piece worth of metal in our bodies, of these transition metals, a very, very small amount. You'll see that iron is the most common, and then zinc, and then all the others are at very low concentrations. The essential metals were first discovered in agriculture, so people realized that if their soil didn't have copper or molybdenum or chromium or manganese or vanadium or cobalt, the plants died. And then subsequently they realized that these are also essential for human survival. So in general, these essential metals exist in the body, not in metallic form, obviously. We don't have deposits of metals in our body. They exist as ions, and in general, they act by binding to proteins, and they give the proteins new functions. So here is one example. I'm just showing you the two metals in the protein. There's a whole huge protein around this, which I'm not showing. This is an enzyme called superoxide dismutase. We need it to be functioning, otherwise we will not survive. It catalyzes the breakdown of the harmful superoxide ion, which causes damage. It's one of the main causes of aging. And the copper and the zinc are very important in this enzyme. In fact, if we don't have copper and zinc, the enzyme doesn't behave normally. One of the forms of amyotrophic lateral sclerosis, or ALS, which is quite common. Michael J. Fox has really championed the cause. One of the causes of that is actually that the superoxide dismutase can't hold its copper and zinc, and therefore it can't function anymore. So in this case, the copper is the thing that actually breaks down the superoxide, and the zinc is there to hold the enzyme in the right conformation, in the right position. If the zinc's not there, then the copper falls out and it can't behave the way it should. And here's another example of an important enzyme, or protein, not in us, but in arthropods and mollusks, so things like lobsters. You might have heard that lobsters have blue blood, and this is the reason. Because they don't use hemoglobin to carry their oxygen, they use hemocyanin. And hemocyanin contains two copper compounds, which then bind to the oxygen, and that's what's responsible for carrying the oxygen in the body. And in the same way, we use hemoglobin, which is an iron-containing enzyme, to carry the oxygen. And these proteins, we always think of proteins as organic molecules made of carbon and hydrogen and nitrogen and oxygen, but in fact, over half of proteins contain metals. They won't function without metals there. So essential metals are really important to us. If we don't have enough essential metal, we will not survive. And this is a dose response curve, which basically says this is how much of the metal we have, and this is the effect, a positive effect or a negative effect. So we've already seen that if we don't have enough metal, we won't survive. We need to supplement it because we can't function normally. We then have this concentration window here where we have normal function. But then at very high concentrations, we also have bad effects. So we're going to later talk about toxic metals, but the truth is that all metals could potentially be toxic, and our bodies need to really tightly regulate their concentrations. Oops. So even essential metals at high concentrations will be toxic. So I'm going to tell you a little bit about iron as an example of an essential metal. It's the most abundant essential metal we saw before, and its main role in the body is carrying oxygen in hemoglobin, but it has many other roles. So if we have too little iron, then we don't get good oxygenation of essential organs. We get very tired very easily. That's one of the first symptoms of low iron. And of course, anemia is the name of when we don't have enough iron. We are not able to carry oxygen efficiently. But if we have too much iron, also we will have toxicity. So to treat iron deficiency, we need to take iron. And basically, iron supplements are just iron salts that we take, or we're told to eat foods that are rich in iron. But I think one interesting thing is that if any of you have been on iron supplementation or someone in your family has been, you're told a few rules about when to take your iron supplements. And there's some really simple chemistry behind this. So the first thing you're told is to take iron supplements with a glass of orange juice. Does anyone have an idea of why that might be? Yep.
Vitamin C helps you.
Yep. Do you know what is in vitamin C? Yeah. That's right. So vitamin C is ascorbic acid, and ascorbate is an ion that can bind to iron and help it to be absorbed. Okay, you're told not to take iron supplements with a glass of milk. Any ideas about that one? Calcium? What do you think the calcium's going to do? Okay, so calcium is another metal, so it's not going to bind to the iron, it's actually going to compete with the iron. So it will stop the iron being taken up because it will be taken up instead. The other thing, what else does milk contain? Yeah, casein or proteins, right? And those proteins are good at binding to metals, and so they will bind to the iron and stop the iron being taken up. You're also told not to take iron supplements when you have a bacterial infection. Any ideas about that? Yeah? Yes, it could accelerate their growth because we've said that metals are essential for life. Metals are really essential for bacteria to survive, and so we're basically feeding them with the iron that they need in order to survive. And so that's why you're told not to take iron supplements. So that's how to treat too little iron. If you have too much iron, it can also be toxic. And the reason for that is that iron is really good at, well, we know that if we have an iron nail, it will rust. It's really good at undergoing redox reactions, oxidizing and reducing. And in the body, the redox reaction it takes place part of is the Fenton reaction, where it reacts with hydrogen peroxide and it produces this free radical, the hydroxyl radical. And free radicals that have an unpaired electron, they want to get rid of that unpaired electron, so they will react with whatever they find next in the body. And often that is DNA, which causes DNA damage, potential mutations, or it's proteins, which cause breakdown of the function of the cell, or it's lipids, which cause the cell itself to break open. So this Fenton reaction is highly damaging in the body, and this is why we can't have too much iron. So there's actually a genetic disease called hemochromatosis. It's the most common genetic disease in Celtic people, so people from Ireland and Scotland and the north of England. And the reason that they think that it's so prevalent in that population is because when the Romans invaded Britain, they pushed the Celts out to Scotland and Ireland, and hemochromatosis gives a benefit. One benefit it gives is it allows women to carry their babies to term so that they're older when they're born. And so under this huge stress when they were being chased out into the very cold areas, those babies were the ones that survived, the babies that had gone to term because their mothers had hemochromatosis, and so that allowed that gene to persist in the population. And so that's why today the Celtic population tends to have a higher level of hemochromatosis than anyone else. And it's quite common that now people are discovering if they've got a family history of heart disease or of diabetes, it's actually because of this stress that's caused by the Fenton reaction because they have hemochromatosis. Does anyone have any thoughts about how you might treat it? How might you get rid of excess iron? Yep? Yep. Yeah, great. So that's actually the simplest way to treat it. You just take blood regularly, and that's depleting the iron in the hemoglobin. That's forcing the body to use its other iron stores to replace the blood. And so that's a pretty simple treatment for this genetic disorder. So we were very interested in studying the Fenton reaction in cells. And so we talked a little bit about this yesterday. You all preempted my this talk today because we talked about conjugation in molecules. And remember, conjugation is alternating double and single bonds. This molecule here is called rhodamine. It's similar to the fluorescein we saw yesterday, but we've got nitrogens at these positions. Rhodamine is a red dye. It fluoresces red. And so we've said that when a molecule is more conjugated, we have more fluorescence, that in general also longer wavelength fluorescence. So rhodamine can exist in a couple of different forms. Here is the ring closed form here, and here is the ring open form, where you can see we don't have this ring here. And can you tell me which one of these two is more conjugated? The one on the left or the one on the right? So the one on the right, because if we count through how many bonds we have that are alternating double and single bonds, we have 1, 2, 3, 4, 5, 6, 7, but then we can't get to the next one. We don't have another double bond next here. Whereas here we can go all the way through the molecule and we have alternating double and single bonds all the way through. So this ring open form is fluorescent and the ring closed form is non-fluorescent. And so we can use this to build a sensor, because if we can build it so that whatever we want to study causes this ring opening, then we go from a fluorescence off state to a fluorescence on state. And so actually all the examples of fluorescence sensors I'm going to show you today use this mechanism. There are lots of other ways to change fluorescence, but I'm just going to focus on this way. So this is the molecule that we made. This is my former PhD student, Zhanglong Wang, who has gone back to China since he finished his PhD a couple of years ago. But he made this sensor here. And you can see that in the absence of iron, it's in the ring closed form, so it's not fluorescent. But when iron comes along, in the presence of hydrogen peroxide, so this is why we're studying Fenton chemistry, because the Fenton reaction is iron plus hydrogen peroxide, it actually causes the breaking of this bond, and we now get this form here, which is a ring open form and therefore fluorescent. And so We can see this. This is the spectrum that we see. The blue is the probe on its own, and here the red is when we treat the probe with iron and hydrogen peroxide. And we can put it into cells and do some cell studies. So here you can see, well, you can't see the cells because there's no fluorescence coming from them. But when we treat them with both iron and hydrogen peroxide, we get much brighter fluorescence, much brighter than when we treat them with iron alone or with hydrogen peroxide alone. So that's a bit of a story about our sensing iron. I'm going to move on now to talk about our toxic metals and why we might want to sense them. So this is the dose-response curve for a toxic metal. That is, under no circumstances does it have a positive effect on health. There is a region where we can tolerate toxic metals, and then at every other concentration it has a negative effect. So this is the toxicity periodic table. These are the metals that we generally worry about. We call these the heavy metals. So silver and cadmium and mercury and lead are the things that are most often talked about. We also have a whole lot of compounds in green which would be potentially toxic, but they're not soluble enough for us ever to encounter them at high enough concentrations to have biological effect. And in fact, the only times they ever really have effect are in cases where people have kidney disease and therefore can't get rid of these metals normally through urine, and then they will accumulate and may have effect. Chromium is here. It's actually an essential metal. We already looked at it as an essential metal. Whoops. But chromium can cause cancer, and so that's why it's here. It depends on what oxidation state it's in, on whether it is essential or whether it will cause cancer. And so if we have a look at the top 20 hazardous substances defined by an agency in the US, you'll see that these inorganic compounds, arsenic and lead and mercury and cadmium, are right up the top. So they're really, really important, and it's really important that we control exposure. So I thought I'd tell you today the story of lead. Lead is an interesting story. It's one of the first toxic metals that humans encountered because it's been mined since 6,500 BC. And it was used heavily during Roman times. They made lead piping. That's why, of course, the word plumbing comes from the Latin plumbum, which is the Latin for lead, and that's why the elemental name for lead is Pb. They used lead acetate to sweeten their wine. Their wine was blue because they put this lead acetate salt in it. They used lead oxides to whiten their complexion. Now, we can sit here and say how silly the Romans were for doing this. They had absolutely no idea of the effects on their health. We can talk more about this later if you want, but we maybe are doing the same thing now, right? We let a lot of nanoparticles now be regulated or be put into the market without regulation. Titanium dioxide we now use as a whitening agent in our sunscreens and our cosmetics, and in the white color on M&Ms is titanium dioxide. And it's now been banned by the EU because they think there may be negative health effects. So we may be going through exactly the same thing that the Romans were going through, where we think that we've got this new amazing chemical, we put it into lots of products, and then we realize that there are effects. There is talk, of course, that the lead poisoning was the cause of the downfall of the Roman Empire, and I'm sure there were many political things that led to it, but certainly it did have bad effects on them. So if we look here at the production worldwide of lead, you can see this is 5,000 years before present, or before 1980 when this paper was published. This is when they first discovered a really simple method of extracting lead from ores. This is when they introduced coins, and therefore they needed to extract lead to make their coins. And you can see that in the Roman period, they were producing a huge amount of lead. There came a point when they exhausted all of the lead in their mines, and the concentrations decreased again. But then, since the Industrial Revolution, lead production increased again. But it's really important to emphasize that lead was only introduced about 5,000 years ago. And so therefore, we haven't had time to develop the proteins. Evolution hasn't had time to come up with ways to get rid of lead or to deal with it. And that's why it's toxic. So lead, interestingly, is the oldest recognized toxic metal. In the 2nd century BC, the Greek poet, botanist and poet Lysander described the poisoning associated with lead. And I think one other interesting thing, and again, this is early chemistry that was happening, was that people would be told to consume butter as a remedy against lead poisoning. So this is a picture here from an old textbook from 1556 on things to do with metals. And you can see here is the smelter where they're actually making the lead from the lead ore. And then in the front here is someone eating butter. So we've already sort of talked about why milk has effect on metal uptake. So what are some thoughts about why we might want to take butter to counteract lead poisoning? Yep.
The proteins and other compounds might form into the lead and it's not as effective as deadly.
Yes, perfect. So the proteins in the butter or in the milk will bind to the lead and therefore maybe prevent it from being taken up for exactly the same reason that it prevents iron being absorbed. What else? So what else does milk contain? We've already talked about calcium, okay, and so here we're actually giving the body calcium, and we'll see in a minute that one of the things lead does is it actually gets rid of calcium in the body. So we're flooding the body with calcium to prevent it from being able to be poisoned by lead. I guess why they're not just drinking milk and why they're having butter is probably because of the fat, and I think the fat also will mean that the lead won't be taken up, it won't be absorbed, it'll just be passed through the body. So I thought that was interesting, that they obviously had no idea of any of the science behind this, and yet they still had worked out that butter could somewhat help treat lead poisoning. Obviously, it's not a treatment today. So one of the reasons that lead is toxic is because it can bind to proteins in the body and stop them from acting. And in fact, what it mainly binds to is the sulfur atoms in proteins. And sulfurs are very important in proteins because that's what holds the protein structure together. And so if lead and this is true of all heavy metals, they bind to the sulfur in proteins, disrupt the protein function, and therefore the cell cannot behave normally. The other thing lead does is that it mimics calcium. And because the body didn't encounter lead during evolution, the enzymes that take calcium into the cell can also take lead into the cell. They didn't evolve to be selective for calcium over lead, and so therefore they don't recognize that lead is any different from calcium. They take up lead. So this is an X-ray of a patient who was exposed to a high level of lead early in life, and you can see that they've actually deposited lead in their bones rather than calcium. And because lead is heavier, it's more dense, and so that's why we get these white regions of greater density. And that's bad because then the bones are more brittle and they can break much more easily. So that's the reason lead is toxic. We are fortunate today that we very rarely encounter lead. We've obviously banned lead in petrol and lead in paints. There are some cases where people are concerned that there's lead piping in houses, but in general, we don't encounter lead in the same ways. But we still want to understand it, and we want to understand how it gets into cells and how it interacts with the body. So this is another former PhD student, Jianping Zhu, who again has finished with me. She's now doing some postdoctoral work in the US. But one of the things she worked on her project was making a sensor for lead. So here is her sensor. Again, you can see that in the absence of lead, we're in the ring closed form, so it's not fluorescent. When lead binds, it binds to this nitrogen here, and that causes the ring to open. And so now in the presence of lead, we have an opening of the ring. And again, this is a rhodamine compound, so we have this red fluorescence. So one thing that is very important when we sense molecules is that we want our sensors to be selective for the chemicals we're interested in. And so this is a selectivity plot where we've measured a whole lot of different metals with the sensor, and you can see that it responds very well to lead. It also responds a little bit to cadmium. That's okay because we don't have cadmium in our cells, and so we don't expect that we will have high levels of interference, but we do need to be really careful when we look at sensors to see what it is that they're actually responsive to. So it's a bit hard to see. I'll turn the lights down again. So it's a bit hard to see, but these are cells under a apparently this does not change anything. These are cells that have been treated with our probe, and you can see there's no fluorescence. And then when we pre-treated cells with lead, you can see we have a turn on in fluorescence in the cells. The question we wanted to ask is, how is lead taken up into cells? Things can either cross the cell membrane passively, or they can be taken up by pumps which actively accumulate it into the cell. And this is important because if there are pumps that are good at accumulating lead, then maybe that's a way for us to stop lead being taken into cells. And the way we do this experiment is that we look at the uptake at 37 degrees and then the uptake at 25 degrees, because the body needs to operate at 37 degrees, so all of the proteins and enzymes will operate, all of the pumps will work at 37 degrees, and when we cool it down to 25 degrees, there will be no pumps that will work. The only thing that will happen is passive diffusion. And it's a bit hard to see, but the only time we see fluorescence, this is the fluorescence image, this is just the cells under a regular light microscope, and this is the overlay. The only time we see fluorescence is when we're at 37 degrees. So that tells us that lead is being actively transported into the cell. That's the first time that this had been discovered. And so now we maybe want to look and try and find out what the enzymes are that take it into the cell, and then we can see whether we can come up with ways to inhibit that to treat lead poisoning. Okay, so that's a story about toxic metals. Now we're going to move on to talking about therapeutic metals. Now theoretically, as I said, anything can be incorporated into a molecule that might have therapeutic applications, so both toxic and essential metals are used in therapy. But the story I want to tell you today, here's the therapeutic periodic table. So you can see in yellow, these are all of the metals that are used in inorganic pharmaceuticals, Then we also have a role for radionuclides. So if we have the radioactive version of the element, that can be used in therapy. And then there's also cases where we can have molecules that absorb radiation and it's concentrated and have a higher effect. And so gadolinium is an example of a metal that does that. But we're just going to focus on the inorganic pharmaceuticals. And I just want to tell you the story of the first discovered inorganic pharmaceutical, which has been really important in the field and in the treatment of cancer in general. So this is a story, it's often used as a story of serendipity in science, where you make a discovery accidentally. But I think it's actually a story of someone who was a brilliant scientist, who came up with an unexpected result and didn't just leave it, but he pursued it, and that caused huge impacts across medicine and chemistry. So this is Barnett Rosenberg. He was a biophysicist, and he just had a thought that cells, when they're undergoing division, have these regions here between the cells. This is the DNA obviously lining up. And that looked somewhat similar to an electric dipole. So he thought, maybe I'll be able to observe this electric dipole-like effect in cells. And I think his idea was, if you take iron filings and you put two magnets, you will end up with a picture that looks like this, where the iron filings are lining up between the two magnets. So he thought that he could do this by taking two electrodes and putting them into a solution containing E. coli or bacteria. And he thought that when he came back the next morning, he would see the E. coli beautifully lined up between the two poles of these two electrodes. That's not what happened when he came back the next morning. What he found was that the E. coli had stopped functioning normally. They'd stopped being able to divide normally. They'd actually become really elongated and they were starting to die. Now, he could have just left this experiment here. He could have just said, oh, that didn't work. Maybe that was a silly idea. I won't continue. But what he did was he was determined to work out what it was that caused this, what it was in the solution that caused these bacteria to behave in such a funny way. And he thought to himself, we've got platinum here from the electrodes, we have ammonium and we have chloride. And so he drew out the possible compounds that could arise from platinum and ammonium and chloride. These are only a few of them. We can draw out many more examples. But he then tested them to see which of these had the effect. And what he discovered was that it was this compound. So this compound is called cis-diamino dichloroplatinum, but we normally call it cisplatin. Cisplatin was actually already reported. Barnett Rosenberg was not the first person to record it, but what he did was discover that it didn't just prevent replication in E. coli, it also killed cancer cells. So this was the first inorganic compound that was approved by the FDA in 1978, and it proved to be highly effective against testicular and ovarian cancers. And just to show you some graphs, this is the survival rate against testicular cancer in 1970, and now it's almost 100%, and that is because of the introduction of cisplatin in the treatment. Ovarian cancer has had much lower survival rates. It's one of the most devastating of the cancers, below 20%, but since the introduction of cisplatin, it's almost doubled the survival rate. So cisplatin has been hugely, hugely useful. It's used in Australia in over half of all chemotherapies. Not on its own, normally in combination chemotherapy, but that's pretty amazing for a pretty small molecule to have such wide effect. And it's really started this area of bioinorganic chemistry that I'm part of now, because people realized that metal complexes could be really valuable. So the way it works is that it goes into DNA and the platinum binds to the DNA, it goes into the nucleus and the platinum binds to the DNA and makes it bend. And this bent DNA can't function normally. The cell tries to repair the bent DNA. It sends these proteins here to go in and try and repair it. They fail because the platinum binds very strongly to the DNA, and therefore the cell signals apoptosis. It signals itself to undergo cell death to get rid of these genetic mutations. The idea is that cisplatin will act on any nucleus, right? Why just cancer cells? But because cancer cells replicate most often, they're replicating really fast, they therefore have more DNA activity, and so the idea is that cisplatin will have more effect on cancer cells than healthy cells. That doesn't really work, and cisplatin, in fact, has really bad toxicity side effects. So the things we hear about as side effects of chemotherapy, like hair loss, sometimes hearing loss, are generally the results of cisplatin treatment. The other problem is that cancer cells can come up with ways to get rid of cisplatin, to actively pump it out or to stop it from having effect, and so they can become resistant. So we've been interested in a number of questions. One is that only 1% of cisplatin actually gets to the nucleus. So either it's really inefficient or it's doing other things in the cell that we've just not discovered yet. And so we do a lot of research in this area in trying to understand what else it is that cisplatin is affecting. In fact, we've discovered that cisplatin changes iron levels in the cell and copper levels in the cell, and maybe that's how it's killing cancer cells. And the second question we wanted to ask was, how can we control the levels of cisplatin so that they're not so toxic, so that when a doctor gives the dose to the patient, they're giving the right dose? So this is one thing that Clara Shen, a former PhD student in my group, in fact, she did this in her honours year. So just after her undergraduate degree, she did this work. She made this sensor. This is a fluorescent molecule, which we saw yesterday, very similar and still works on this ring opening mechanism. So it's ring closed, and then when platinum binds, it opens the ring and turns on the fluorescence. And we get a good response to platinum over all other metals. But not only is it responsive to just platinum, it's actually only responsive to one form of platinum, which is this particular compound here. And this compound is actually a metabolite of cisplatin. So what we're measuring is this sort of compound here, where platinum is partially bound to DNA, but not completely bound to DNA. So we have a sensor that tells us how effective the platinum is being. Is it starting to be metabolized in the cell and bind to DNA? And here you can see we get greatest fluorescence after 1 hour and 6 hours, and then by 24 hours, the platinum is all bound to the DNA, no longer has any effect. So this and lots of other tools we've made in our group are starting to let us understand how platinum interacts with cells. But I'm going to move to another area, a different type of sensing. And that's to do with this question of dosage control. So I've said platinum's really important. It's used in more than 20 different cancer types. Worldwide, 40% of chemotherapy patients receive platinum, but 73% of those patients get the wrong dose. They have such high dosages that they have really bad toxicity and they have to actually stop treatment. And so we wanted to come up with a better way of measuring platinum levels in the blood so that patients wouldn't be given the wrong dose. So to do this, we needed to operate on a different sort of sensing mechanism, and we were inspired by the way our noses work, because we can distinguish many, many different types of smells. We can distinguish thousands of different smells, and we know this because smell relates to taste. We can taste so many different types of foods. But in our brain, in our nose, we only have 400 different smell receptors. And we can smell far more than 400 smells. In fact, we can distinctly smell more than 1 trillion scientists tell us. And the way we do this is by using combinations of the different receptors to give us those distinct differences. And so that's what we wanted to do. We wanted to measure many things at the same time by using lots of receptors. So instead of 400 smell receptors, we've now got 6 fluorescent sensors. This is work by Linda Mitchell, a PhD student who recently finished. And so she built these 6 different fluorescent sensors, and now she can measure lots of different things at the same time. We're not measuring smells, we're measuring metals or metal complexes. And so what that gives us is a really complicated pattern where we have every sensor against every metal, and we can't look at this pattern and understand what's going on, but fortunately we can use computational power to look at these complicated patterns and work out what's going on. And so we can take those many dimensions, this is how many dimensions of data we have, and our statistical software can reduce it down to 2 dimensions of data, and now we can see that each of our different metals is very well distinguished from each other. So we can use this complicated sensor array to tell us what is platinum and what is cobalt and what is iron. We can also use it to tell us what is, and I haven't shown you here, but we can also use it to tell us what is active cisplatin in the body, and we're starting to use it to tell us what is the concentration of cisplatin in the body. So this is a test that we're now developing. We've taken into clinical trials, and we're working with doctors to work out how we can actually use it to measure the platinum levels in patients in a way that has never been able to be done before. So that's just a bit of the end of my quick story about metals in health. A recap, transition metals are key to human health. Some of them we need to survive. Some of them kill us. Some of them have therapeutic applications. But we can use fluorescent sensors like what we do in our lab to help us understand more about these interactions. I think that's it.